In this lecture, we'll talk about transgenic animals. So, how to create transgenic animals and what is their utility. So, we'll go through this in this lecture. A transgenic animal is one that carries a foreign gene that has been inserted into its genome. It is a process by which mixing of genes takes place. Foreign genes are inserted into the germline of the animal so that it can be transmitted to the progeny. Transgenic technology has led to the development of fishes, livestock, and other animals with altered genetic profiles, which is beneficial to the mankind. First transgenic animal was a super mouse, which was created by Rare Brinstead from University of Pennsylvania, Richard Parmiter, University of Washington in 1982. It was created by inserting a human growth hormone gene in mouse genome. Because of the human growth hormone, overexpression of human growth hormone, size of the mouse became very big, very large. So it was inserted into the mouse genome. The offspring was much larger than the parents. So mouse is very common for creating transgenic experiment, but other animals including pig, goat, cow, sheep, fish have been now generated. So these animals have been made transgenic now. So what are the methods to produce transgenic animals? The common methods are the embryonic stem cells method, pronuclease method, or retrovirus mediated gene transfer. So these are the three methods which can be employed to create a transgenic animal. Embryonic stem cell method. In this, embryonic stem cells are collected. Formation of gametocytes, injection into blastocysts, and then injection into the foster mothers, and then we get a transgenic animal. So this is using embryonic stem cells. Pronuclease method. Once you prepare the DNA, fusion of once you have the foreign gene vector available, you fuse with the male pronuclease with the desired DNA. Formation of diploid zygote. Then mitotic division will occur, formation of two-cell embryos, and then once the two-cell embryo is formed, it's transferred to the foster mother, and new transgenic animal will be born. So this is the pronuclease method. So the method one you can see here. So once we collect the embryonic stem cells here, select, and then you introduce the foreign gene here in this embryonic stem cell. Select for the cells expressing desired genes. So embryonic cells with the foreign genes, then they are injected. So these are then injected into the blastocyst in the inner mass, and then these are implanted in the uterus, in the foster mother, you can say. So test offspring for the presence of gene. Then one has to look for the offspring which has now acquired the foreign gene, which is transferred from the embryonic stem cell. So this is the embryonic stem cell method, this particular one. So male heterozygous offspring to produce homozygous transgenic strains. Once you get the homozygous strain having two copies of this foreign gene, then we will have a transgenic animal. So this is the method one, using embryonic stem cells method. In the second method, we have the desired genes. We can put in male pronucleus, and then we'll get fertile with the egg. And once after fertilization, is implanted in the uterus of the foster mother, and then we'll test for the offspring for the presence of the foreign gene. So this is pronuclease method. Another method is retrovirus mediated gene transfer. It was used as vector to transfer genetic material to the host cells, resulting in chimeras on organ consisting of tissues or parts of diverse genetic constitution. So this is another method, but the first two methods are the commonly used methods. So but this retrovirus also can be used for generating transgenic animals. So this is the steps, various steps for production of transgenic animals. Various steps. The first step is important construction of a transgene. So transgene, For construct transgene, one has to select for the gene of its of interest, any foreign gene, and under a suitable promoter which will be explained in that particular animal. Okay, so this is gene under control of a suitable promoter. Then this gene has to be introduced. So pronuclear microinjection method or embryonic cell stem cell method are commonly used for introduction of gene. So for in case of microinjection method also, a female animal is super ovulated and eggs are collected. The eggs are fertilized in vitro. The transgene containing solution is injected to the male pronucleus using a micropipette here. With help of the micropipette, this can be introduced into the male pronucleus. So here is a female pronucleus here. Here is a male pronucleus. So here it is. So the transgene with containing the harboring the desired gene is injected into the male pronucleus using a micropipette. Eggs with the transgene are kept overnight in an incubator to develop to a two-cell stage. Once the gene is introduced to the male pronucleus, it is put in an incubator to develop into two-cell stage. The eggs are then implanted into the uterus of a foster mother. So that was the pronucleus method. In the embryonic cell method, transgenic animals can be created by manipulating embryonic stem cells. Embryonic stem cells obtained from the inner cell mass of a blastocyst. So inner cell mass, so that one can collect embryonic stem cells. Transgene incorporate then the transgene is introduced into the embryonic cell by any of these methods: microinjection method, retrovirus method, or electroporation method. So any of these methods can be used to introduce transgene into the embryonic stem cells. So after introduction of foreign gene in embryonic stem cells, transgene stem cells are grown in vitro, and then embryonic stem cells with the transgene are selected, and then they are inserted into blastocyst and implanted into a host uterus to grow normally. Then, so these now so embryonic stem cell genetically modified manipulated embryonic stem cells are then introduced into the blastocyst and then implanted into a foster uterus of the foster mother. So this is how the microinjection is carried on. The microinjection of the is carried out in the blastocyst. Microinjection of the embryonic stem cell, genetically manipulated embryonic stem cell into blastocyst. So once you have introduced genes via embryonic stem cells or through microinjection to pronucleus, nucleus, then screening for the transgenic is carried out. So transgenic progenies are screened by PCR. One can use PCR method. This is fast and more accurate to examine the site of incorporation of the gene. So the screening is done using PCR. So one can, by using PCR, one can select for the transgenic animal. Then further animal breeding is done to obtain maximal expression. So then once heterozygous offsprings are made to form homozygous strain. So you have two copies of the transgene, so that we have more expression. To get maximum expression, then breeding is carried out, and you can have the progenies harboring the transgenes. So this is the same thing here. Transgene injection into the mouse eggs, then you have in the uterus surrogate mother, and then you have a transgenic progeny, transgenic mouse basically. So some few problems can occur, like multiple insertion. Then it may cause too much of protein, or it may cause some insertion into essential gene also, which can be lethal, or it can be lethal for the progeny. Insertion into a gene leading to gene silencing. Insertion to different area can affect the gene regulation. So one has to take care of these problems when developing transgenic, so that we don't encounter any of these problems while making transgenic animals. So we need to basically know where the gene is getting inserted in the genome. So there are some example of transgenic animals which have already been developed, like superfish. In the superfish, increased growth and size, growth hormone gene inserted into the fertilized eggs. So one can see the enlarged size of the fish after introducing growth hormone gene. So you can see a very large size of the fish as compared to the wild type one. So transgenic salmon grows about 10 to 11 times faster than normal fish. So they grow much faster also. So you can get uh, fish on edible fish for in a very short span of time in aquaculture, because they grow faster as compared to the wild one. Glowfish, so basically these are the ornamental fish. These are jointing freshwater zebrafish produced by integrating a fluorescent protein gene from jellyfish into embryo of fish, or sea anemone, the red one. So, these are some of the example of transgenic fish which have been developed and already been marketed. Transgenic mouse is one of the common uh, animal for generating transgenics. So, for in case of transgenic mouse, several disease models have been developed, like Alzheimer mouse. In the brain of Alzheimer patient, dead nerve cells are entangled in a protein called amyloid. Mouse made by introducing amyloid precursor gene into fertilized egg of the mice. So you can have a you can develop Alzheimer model for Alzheimer disease. So one can test various drugs um, for targeting Alzheimer disease, actually for as treatment of Alzheimer disease. So they can be tested on this Alzheimer mouse now, because they are making the same protein. So these are very excellent model for testing Alzheimer drugs. Oncomouse, mouse model to study cancer, made by inserting activated oncogenes. So oncogenes are introduced into the mouse genome. So now you have the oncomouse, in which various drugs can be now tested. Smart mouse, biological model engineered to overexpress NR2B receptor in the synaptic pathway. So this makes the mice learn faster than like juvenile throughout their life. So they become very smart. So they learn very fast. So just by overexpressing NR2B receptors. So this is a normal mouse. So this is another one in which green fluorescent protein has been introduced into the mice. So here is a normal mice. Yeah, young one of normal mice. Here you can see the transgenic mice, which are glowing green in color. Transgenic pig and enviro pig, actually now transgenic pig has been also created. Pigs have a trouble fully digesting a compound known as phytate, found in many cereal grains used to feed them. So they are unable to digest this because of phytate presence in the cereal grains. So transgenic pigs are created by introducing phytase gene in E. coli. Phytase enzyme is thus produced in salivary gland of the pig. It degrades intentional phytate with the release of phosphate, and that is easily digested by pigs now. So once the phytase gene is introduced in the transgenic pig, it will degrade the phytate, and then it will be cereal grains can be easily digested by the pig. And the pig can be used for, or because pig for the organ transplant. Transgenic pigs have been developed for obtaining organs for transplanting in humans, actually. Pigs with human genes, in order to decrease the chance of organ rejection by the human body. So pigs organs can be used for transplantation in human. So but to prevent the rejection, some of the genes, human genes, can be introduced in the pigs. So those pigs, then pig organ can be used for transplantation in human body. So this work also is going on. So this is very important work for doing for transplantation in human, wherever there is a requirement for organ transplant like liver, kidney. Okay, so these so this pigs with the human genes. So human genes can be introduced in the pigs so that their organs may be more very similar to the human one, so that won't be get rejected by the human body then. So this is another work in which transgenic animals can play a very important role. Particularly transgenic pigs can play a very important role. So beside this, transgenic rabbit has been developed, like in this green fluorescent protein has been introduced into the rabbit. Transgenic fish, tilapia, salmon, catfish, they have been made transgenic now by introducing growth hormone gene. So they grow up to 6 times faster than the wild type. So this one transgenic fish have been there. Transgenic cow has been also developed. Transgenic cows carrying extra copies of two types of casein gene produce 13% more milk protein. So they have extra copies of this transgenic cow has extra copies of the casein gene, extra copies of casein gene. So they produce 13% more milk protein. So they are more nutritious as compared to the wild type. So other animals have been now, several different animals now we have been genetically modified, like mice, goat, sheep, chicken, cow, horse, dogs, fish. So these are some of the example of animals which have been made now transgenics by different groups. So this is transgenic sheep. Tracy is the first transgenic animal to produce a recombinant protein in her milk. So transgenic sheep is also, besides this recombinant protein in her milk. So this transgenic sheep has been used for studying is a very ideal model for studying immunology, human blood clotting factor, transplantation, hematology, biological products manufacturing, recombinant DNA, drug production in milk. So one can use this transgenic sheep for producing drugs or recombinant protein for therapeutic purpose. So there's so many advantages of these transgenic animals now. So now transgenic livestock can be used as a bioreactor. So where cells have been engineered to synthesize marketable protein, means bioreactor means they can large amount of recombinant proteins can be produced using these transgenic livestock for human use. This will be more economical. So transgenic animal will be more economical than producing desired protein in animal cell culture, because animal cell culture is expensive. So it will be more economical to make these recombinant proteins in the livestock, particularly like cattle. Transgenic cows are made to produce protein lactoferrin and interferon in their milk. So once you make these transgenic cows, one can get the recombinant protein from their milk easily. So you don't have to sacrifice animals. You can just collect the milk and purify the protein through downstream processing. So lactoferrin and interferon have already been produced in their milk. So this prion-free cows resistant to mad cow disease. Prion-free, one can create transgenic cattle which are now resistant to mad cow disease. So that also can be created. Transgenic sheep can be developed for good quality of wool production. Good, if you want, you can modify it. You can have transgenic sheep with better quality of wool. Transgenic goat that could express tissue plasminogen activator, antithrombin 3, spider silk in the milk. So these have been already been done, transgenic goat. So these are the transgenic livestock which can be used as a bioreactor. Transgenic monkey is also there now. Andy was the first transgenic monkey born in 2000. Andy stands for inserted DNA spelled backward. And in gene bias was used to insert the harmless gene for green fluorescent GFP into the Andes rhesus genome. This ND proves that transgenic primates can be created and can express a foreign gene delivered into their genome. So monkey also has been made transgenic. So this. So now we'll go through some of why these transgenic animals are beneficial. What is their significance? What is their importance? So in the field of medical, so they can be used as a disease model for studying cancer or Alzheimer disease or any other disease. They can be used as a bioreactor for pharmaceutical. Various pharmaceutical drugs can be produced, particularly recombinant protein for therapeutic purpose. Xenotransplantation, like in case of transgenic pigs, there are organ which have been modified, in which human genes has been introduced. So those organ from those transgenic pigs can be used for transplantation in human. Agriculture important, like disease resistance animal can be created, like mad cow, um, uh, cattle resistant from mad cow disease. These animal can be created for improving quality and quantity of milk. Milk and their quality and quantity of milk can be improved. Meat, eggs and wool production, this also can be improved using transgenic animals. Industrial importance, toxicity sensitive animals to test their chemicals. So some toxic sensitive transgenic animal can be created to test the various chemical pesticides, drugs and all that one. Spider silk in milk, because spider silk is strength is very high as compared to uh, uh, normal silk. Spider silk can be made in milk of the goat. So one can then easily purify large amount of spider silk. So transgenic got engineered to produce human breast milk. So this is another one. Utility of transgenic goats have been engineered to produce human breast milk. University of California created the transgenic goats by transferring human genes for breast milk enzymes and protein into the goat embryos now. Produce 60% of lysozyme lactate found in the human mother's milk. So it's more like similar like human breast milk. For babies of mother, this is actually useful for babies of mother who are not present or can't nurse them. Milk from these transgenic goat provide the next best alternative. So because it will have important protein from the human breast milk. So this is another utility. So some of the bioluminescent transgenic mouse model can be used for study memory gland tumor for development. So all these, there is a lot of utility for these transgenic animals. So there are several advantages like increased growth rate, improved disease resistance, improved food conversion rate, increased muscle mass, improved nutritional quality, improved wool quality also. But there are some disadvantages also. Inserted gene has multiple functions. Breeding problem. So if there is some breeding issues can occur with the transgenic, so this has to be taken care of. Sometimes leads to mutagenesis, so this also has to be addressed. Low survival rate of transgenic animals. So so this also is another disadvantage. One has to. So this is just one example of transgenic technology as an alternative to drug producing cells. How we can use this transgenic animal as a bioreactor. So this is desirable to have the protein drug secreted in an essentially retrievable manner. So the best use of advantage of using transgenic as drug factories or producing large amount of recombinant protein is that they can be easily retrieved, because this protein will be made, this recombinant protein will be made in the milk of the cattles. So it will be secreted in the milk, so it will be easily retrieved It will be easily retrieved, and it can be you don't we don't have to sacrifice the animal for purifying or obtaining this recombinant protein. So this is a very good advantage of transgenic animals. Like a recombinant secreted in the milk of the transgenic animal could be produced in a large quantities and easily retrieved from the animal, because one can easily collect milk, large amount of milk, and then we can have large amount of recombinant protein. So here does whatever the gene of interest, foreign gene of interest, one has to fuse with the promoter sequence of the major milk protein, like casein protein. It can be the promoter has should be from the milk protein. So then the recombinant protein will be secreted out in the milk of the cattle, transgenic cattle. Although this foreign gene will be present in all of the cells of the transgenic animal, it will be present in all the cell, but it will only be expressed in the mammary tissues only. So the expression will be highly specific. It will be expressed only in the mammary tissues, because we'll be using the promoter sequence of the milk protein. So this is the advantage. It won't get expressed in other tissue cells of the transgenic animal, only in the mammary tissues. So this is one example. Human protein C was expressed. It's a blood protein, function to control blood clotting. Some individuals have inborn deficiency, require exogenous protein C. So this protein C is is required to control blood clotting. So this protein C now can be made in large amount from the cattles. So this is one example here now. So human DNA coding for protein of interest, any like protein C or some any other gene for any desirable trait can be fused with the milk protein promoter, and then fertilize, put in the pronucleus, male pronucleus, injected into the male pronucleus here, and then and then it's implanted in the foster mother. It's the same thing here, or one can use for human sequence of protein of human gene of any of interest, and then introduce the male pronucleus. So genie, the first genetically engineered animal to produce a human protein drug or human protein C in her milk. So this is one transgenic pig which is making large amount of human protein C in her milk. So this transgenic pig, which is called genie, is produce sufficient quantities of human protein C. 1 gram is 1 gram of human protein C per liter of the milk. So you can imagine how much protein is there. This yield is quite significant, because this much of yield is not possible from animal cell culture or E. coli and all that one. So 1 liter of milk, just 1 liter of milk is making 1 gram of human protein C. So this is 200 times more than present in the human blood. So this very good example of making large amount of recombinant protein, which has therapeutic value also. So human protein C. So yield is very good from genie. So there is some other example beside protein C. There is some other therapeutic protein production using transgenic animals, like growth hormone. Human growth hormone is being produced from the goat. Human fertility hormones from cow, from transgenic cow. Similarly, fibrinogen is being made from the transgenic sheep. So these are some more examples. So this is one example where the drug has drug which is produced from a transgenic animal, which has been approved by the FDA for human use. So this protein is made from a transgenic goat. The drug meant to prevent the drug meant to prevent fatal blood clot in people with rare condition is a human protein extracted from the milk of genetically engineered goats. At the same time, the FDA also approved the goat used to make the drug, the first such animal cleared under guidelines the agency adopted to regulate the use of transgenic animal in the nation's drugs and food supply. So this drug is made by a company called, this transgenic goat was made by a company called GTC Biotherapeutics. The human anti-clotting protein is produced by a herd of 200 bioengineered goats living under carefully controlled conditions on a farm in farm in USA. Proponents say that such pharma animals could become a means of producing biotechnology drugs at a lower cost or in greater quantities than the existing batch, which include extract protein from the donated human blood or growing them in large steel vats of genetically engineered cells. So this is much easy, it's more economical, and one can get a large amount of this recombinant protein. So the protein basically is the protein in the goat milk. This is actually antithrombin. Is sometimes in short supplies or available for pharmaceutical use because of a shortage of human plasma donation. So this protein was generally earlier extracted from human plasma, but the amount was very low. But now this antithrombin is being made from a transgenic goat now, and all around 200 genetically engineered goats are they were made by this biotherapeutic company, and so they are now able to get large amount of this particular protein. GTC Biotherapeutics said one of its goat can produce as much as antithrombin in a year as it can be derived from the 90,000 blood donation. And if more drug is needed, the herd can be expanded. So this is already this particular drug antithrombin is which is made from the transgenic goat has been approved by FDA and it is being used in USA. So this is a major milestone in uh, research on transgenic animals. Okay, so thanks.